ChIP-seq is an acronym that stands for chromatin immunoprecipitation followed by sequencing. This video is meant to be a quick overview over the basics of chip sequencing in an effort to vulgarize the concept. Chromatin immunoprecipitation is an experimental technique used to investigate the interaction between proteins and DNA in the cell. It aims to determine whether specific proteins are associated with specific genomic regions. There is two types of proteins that we can be looking at when we are doing chip sequencing, the transcription factors and the histones. The transcription factors are proteins that bind to the genome and interact with the gene. They can enhance and repress a gene and have some other functions. The histones are a group of proteins that are attached together and around which the DNA will wrap to be folded. In the case of histone, we can study the effect of modifications on the folding of DNA. If we take the example of our transcription factor that interact with the gene, we have it at some place over the chromosome. The first thing that we will do is to bind it very strongly to the DNA. We call that cross-linking. After that, we will destroy the cell by sonication. It will cut the DNA that is not protected by the protein. Once the complex protein DNA is floating free in the tube, we can add little beads with a specific antibody attached to it. The antibody is specific to the transcription factor and binds to it. We can then extract the beads, reverse the cross-linking, and get our fragment of DNA to feed them in the sequencer. The sequences of the fragments that the sequencer have been reading and putting into files have to be put in order according to a reference sequence. And then once your sequences are aligned to the genome, you can try and see what are the parts of the genome that have a big pile up. It could lead to a region where your transcription factor actually bound. At the moment, the peak calling needs to be done against the control. If you remember the chromatin immunoprecipitation of the transcription factor, you need to provide the transcription factor to your experiment. The control is the same experiment without adding your transcription factor. That way, you try to eliminate enriched zone that could be an artifact by subtracting the control. As you can see on the example, I drew one zone that is meant to be an artifact and that the software wouldn't recognize as a peak. These kind of experiments give you more insight on the genes involved in the disease you are studying and the importance of some protein in this disease. For example, the Enlighten is interested in the importance of some specific proteins in autoimmune disease, like diabetes. For more information on what the Enlighten is searching about, please consult enlight-10.eu. Thanks for watching.